Good morning. As Chris described, we're going to take a little bit of a different direction from the previous panels yesterday and even the one that you had heard this morning, and move away from some things about the cancer environment, about therapeutics, about how much they cost, and about the markets in which they're delivered. We're going to turn instead to talk a little bit more about some of the microscopic views of cancer, at least at the molecular level and in the areas in which we see significant changes happening in the course of, or in the nature of the way the therapies that will be coming versus the ones we've become used to thinking about. Now, epigenetics is in and of itself a little bit different. It's, I think, people have a pretty good understanding, or at least a conceptual view, of what a genome is and what its structure is, and the idea that things are strung along one relatively long molecule. And epigenetics doesn't have, it has a lot of structure, but it doesn't have that same kind of structure, I think, that people have gotten used to. So what we're going to try to talk, we're going to do a little bit of a tutorial as we go through this, and to introduce not just how epigenetics is going to impact cancer and the kinds of therapeutics that you can expect to see, but also about something about epigenetics generally, because we'll also talk briefly about areas of life sciences and of healthcare in which epigenetics plays even a broader role. Now, about 10 years ago, in 2006, Nature Reviews Epigenetics, Nature Reviews Genetics had produced a series of milestones, summarized a series of milestones that covered the developments in cancer that went from roughly the end of the 19th century to the beginning of the 21st. And interestingly, there were about 24 milestones that had been defined, and they contained many of the things that were certainly hot topics. Tumor suppressors, the effective tumor suppressors, oncogenes were certainly things that were at the near the top of what that list was. And interestingly, epigenetics was also included on that list. However, in the case of epigenetics, they added a tagline of emerging, but essentially not there yet. A lot has happened since that article was published roughly 10 years ago, and today the panel is going to walk us through the kinds of things that have happened over that time period, but more importantly, the things we can expect perhaps for the next 10 years. We've been very fortunate, as Chris described, to be able to assemble a panel that consists of a series of both panelists involved in basic biology and in basic science, but also in 3 companies that are at the forefront of delivering compounds both into the clinic and then ultimately into the market. And so we're going to start today by having each panelist introduce themselves, where they are, what they do, and give their own sense as a brief summary of where they see epigenetics fitting with respect to the various ways in which cancer therapies are being developed. And we'll start first with Bob Copeland from Epizyme.
Thank you, Pat. As Pat said, my name is Bob Copeland, and I am Chief Scientific Officer at Epizyme, a publicly traded a biopharmaceutical company here in Cambridge, Massachusetts. I've been with Epizyme since September of 2008. Prior to that, I was Vice President of Cancer Biology for GlaxoSmithKline. And I think it's an incredibly exciting time for epigenetics in oncology. We are starting to see the first true testing of these modalities in the clinic as monotherapies. Work from our own company, Epizyme, from Constellation, from GlaxoSmithKline, from a variety of different companies testing enzyme inhibitors that interface histone methylation and histone acetylation, and in that way control chromatin structure and transcriptional control. So I think it's a very exciting time. I'm really excited to see this transition to the clinic, again as monotherapies and then eventually as combination therapies as well.
Next panelist is Keith Dionne. Keith?
So Keith Dionne, thanks Pat, CEO of Constellation Pharmaceuticals. And I'm reminded of, when I think about epigenetics, of a conversation I had about a dozen years ago with Eric Lander when I was at Millennium Pharmaceuticals. And we were talking about what was going on with sequencing the human genome. I remember Eric surprised as we went from 100,000 genes to 80,000 to 60 to 40 and ultimately to the 22,000 and change genes that comprise each one of us, asking the question, where does all the complexity come from? And I think we've discovered the complexity comes from the mechanism, the proteins of epigenetics that control how those genes and which of those genes are translated, which ones are ignored, and how they activate in each of the cells of our body. In particular, epigenetics are this set of proteins that write tags on the chromatin, erase these tags, read these tags, that basically say, read me, or translate me, or don't read me, or translate me twice as it goes through. This is the control center inside the nucleus that really determines which genes are being translated, which ones are active, which ones are not active. And that controls fundamental programs like making a cell grow, making it differentiate, leading to different kinds of cells in the body, many of which have to do with cancer, many of which have to do outside of cancer as well. And I think with this control, what we're discovering ourselves, as well as Bob and his team at Epizyme and Nancy and others as well, is that we can now control some fundamental programs involved in the development of cancer. A couple of these just to note, the development or the discovery that we could inhibit MYC, one of the most proliferative oncogenes in all of cancer, by inhibiting a bromodomain reader called BET, as well as in a different direction, a different bromodomain reader called CBP, that oncogenes otherwise considered undruggable, we can now stop the translation and therefore deplete the protein of. In a second area, we've seen the ability to advance or understand that often the bane of our existence, most of the drugs that we develop, unfortunately, the cancer develops resistance to. And some work that Jeff Settleman did a while back here in Boston really demonstrated that often the resistance is developed through epigenetic means as opposed through mutation of the actual underlying genome. And he proved this by doing single cell plating of 1,000 different tumor cells, expecting maybe 4 to 5% of those plates to develop resistance according to mutations. Instead, what he saw was 4 to 5% of the cells in every plate developed resistance, and it was governed through an epigenetic means as the cell responded to the stress of the targeted therapy by adjusting its gene programs to develop resistance against that threat, which then went away once the drug threat was taken away. Final area that we look at as being incredibly exciting right now is the development of epigenetics and the immune system. You're going to hear a lot about utilizing the immune system to fight cancer. What we've discovered, and others as well, is that epigenetics is fundamental to the development and control of aspects of the immune system. EZH2, for example, as well as KDM5, two epigenetic proteins, block the ability of not only resistance, but EZH2 on the area of T regulatory activity, the brakes of the immune system. We've seen the development of EZH2 and DNMT inhibitors to really be able to activate the recruitment of T cells into the tumor bed. Checkpoint inhibitors themselves, an exciting area of antibodies. We're now seeing the ability of small molecule epigenetic inhibitors to also block some of those key checkpoints, PD-1, LAG3, and others. So it's an exciting area, expanding a lot of different areas around that are exciting across cancer therapy. And maybe the most exciting part is the realization that it works. We found that epigenetic proteins are druggable, that we can get bioavailability, that there's safety going into the body, and perhaps most important, that there are people walking around today who wouldn't be without some of the drugs that our company and some of the other companies represented here have started to develop and are testing in the clinic right now. So it's an exciting time.
Okay. Cigall?
Thanks, Pat. I'm Segal Kadosh. I'm an assistant professor in pediatric oncology at the Dana-Farber Cancer Institute in Harvard Medical School, having just established my laboratory about 2 years ago after completing my training at Stanford University. Our laboratory is intensely focused on one class of epigenetic regulators that regulate the 3D architecture, three-dimensional architecture of the genome. That is, they alter the genetic structure such that certain genes can be turned on and off in a timely and appropriate manner. This family of epigenetic regulators has jumped to the forefront of attention over the past several years, owing largely to sequencing studies, as both Keith and Bob alluded to. These sequencing studies have revealed very frequent mutations to the various components of these epigenetic regulators, making them actually the most frequently mutated epigenetic regulator in all of human cancer. So our laboratory studies the structure and function, and ultimately towards strategies toward targeting these epigenetic regulators in cancer as well as other diseases.
Thanks. Jeannie?
Hi, so I'm Jeanne Lee. I'm a professor of genetics at Harvard Medical School and Massachusetts General Hospital. I'm also an investigator with the Howard Hughes Medical Institute. And over the last 18 years, we've spent a lot of time studying the epigenetic regulation by long non-coding RNAs. And we've used this model called X chromosome inactivation to understand how long non-coding RNAs regulate gene expression. And using EXIST, which is a long non-coding RNA that spreads over the X chromosome, so it's one of the sex chromosomes, we've learned that polycomb repressive complex 2, PRC2, EZH2 that you've been hearing about, and many other chromatin complexes will actually get recruited to a specific place in the genome by a long non-coding RNA, and in this case, EXIST. But then we've gone on to demonstrate that PRC2 and other chromatin modifiers will actually associate with an entire family of long non-coding RNAs. We call this an RNA interactome, and very frequently, this interactome could be more than 10,000 in membership. And so in thinking about this, it occurred to us that we can actually leverage the specificity, the ability of these long non-coding RNAs to target complexes to a specific location in the genome, so we can leverage that specificity to design a drug to prevent the recruitment of these repressive complexes. And so to translate this into a therapeutic, we started a company called Rana Therapeutics that some of you might have heard of, together with Jean-Francois Formella of Atlas Venture and Art Krieg, who became our first CEO, to do exactly that, which is to design drugs against long non-coding RNAs to interfere with recruitment of a specific complex. And so Pat charged me with the task of explaining long non-coding RNAs today and why they're going to become more and more important for epigenetic therapies, and I hope to do that if time permitting this morning. But I'll begin by saying that there are only 20,000 protein coding genes in our genome. And I think many will say that we're running out of druggable proteins. And so by contrast, there are at least 10 times more long non-coding RNAs in our genomes than protein coding genes. And you might have heard that only 1% of the genome actually makes protein, and that's true, whereas 99% of the genome is considered to be junk. However, what I want to convince you of today is that junk is actually a treasure trove that we should all be mining for epigenetic therapies. We know now that more than 90% of this junk is transcribed into some kind of long non-coding RNA. And while a lot of this will be transcriptional noise, as some people are fond of saying, it's also become very clear that a lot of these long non-coding RNAs actually have function. And so hopefully, with time permitting this morning, I'd like to give you some examples of that.
Nancy?
Great. I'm Nancy Simonian. I'm the CEO of Syros Pharmaceuticals, which is a private platform and therapeutic company in the gene regulation space. My background is I'm a physician by training. I actually did my medical training at MGH and was an assistant professor at Harvard Medical School and MGH before I left about 21 years ago to go into industry. And I've been very fortunate over the last 20 years to have worked on some really important, innovative drugs that have made a big difference for patients. But my experience in biotech, and I would sort of argue this would be the same across the pharmaceutical industry, is that we were often had drugs or pathways we were working on trying to find the right patient populations. And as a physician, we're trained to understand what's wrong with the patient and then figure out how best to treat that patient. And it struck me that we were sort of had the problem backwards, or we were focused on the wrong thing in terms of how we're developing drugs. And if we could really understand the fundamental basis for disease, we could then be in a much better position to identify the optimal point of interventions, the right targets for the right patients. And it was at the time when I left my role as doing drug development that I understood through interactions with an incredible amount of science coming out of multiple institutions, including partners, that the non-coding region of the genome sort of held the keys to what made a cell the cell it is. So just imagine you have 200 different cell types in your body. They have identical genomes, but they all function completely differently. And that's all because there's a program that controls what makes a cell what it is. A skin cell has a completely different program than a muscle cell. And what happens in diseases, including cancer, are those programs go awry. And by studying that non-coding region of the genome and the programs that you heard about from Keith that control the expression of genes, you get the ultimate insight into what's gone awry. And so my interest in why I decided to come and start a brand new company in this space is that the tools and the technologies are now ripe for us to analyze and exploit this understanding of the non-coding genome in these programs to come up with better therapeutics to help patients and also delivering the right drug for the right patient.
John?
Hi, I'm Johnathan Wetstein. I'm an associate professor at Harvard Medical School, and my lab is at the Mass General Hospital Cancer Center. My lab is an epigenetics lab. We study basically how your DNA is packaged in that microenvironment, and we have two goals. One is to understand how we can harness that to optimize either traditional or novel therapeutics that many of my colleagues on the stage are discussing, while at the same time, we're trying to understand and have begun to really delve into understanding how epigenetics, or how you control that DNA microenvironment, is actually impacting drug resistance. How is it affecting that tumor heterogeneity that occurs? And what we've recently discovered is that the process of the heterogeneity, or the process by which epigenetics influences this, is a very directed process. And one of the big challenges, and you heard it yesterday, you've heard it, you'll hear it today, you'll hear it tomorrow, you're going to hear it consistently in the literature over and over again. An Achilles heel for oncology is the ability of a tumor to adapt and change. And so what I'm here to tell you is that the context of epigenetics is an area that controls this. And by having kind of started to resolve those pathways involved in this biologically, what we've been able to do is start to index novel candidates to inhibit so you can suppress the effects, or begin to understand which ones, when you start to touch them, will generate the heterogeneity. So what our goal is in my lab is to kind of bring those two worlds together. Can we identify a way to use them in combination to optimize the therapeutics, new and old? But more importantly, can we target those specifically to suppress the resistance that ultimately is a problem for the patient, or the heterogeneity that's emerging in tissue?
Thanks. And so, having set the stage with respect to now understanding the areas of investigation regarding the epigenome or epigenetics with respect to the development of novel therapies, we want to dig a little deeper into what some of these concepts mean, because at the end of the day, you still need to develop a compound that hits some target and causes some appropriate effect. And so the process by which that happens is obviously non-trivial. We want to dig a little deeper into what's, number one, how some of those things get done, but more importantly, what some of those novel targets are, targets that haven't yet been investigated very much. So I'm going to ask a series of questions, and the first one, Cigall, is for you. Chromatin packages DNA into a very organized, exquisitely organized, three-dimensional canonical architecture that allows a large molecule to be put into a very, among other things, a very small space. But it's recognized that remodeling of the chromatin occurs in different cell cycles, and this remodeling is governed by a series of complexes that, until fairly recently, have not really been well understood. And I think, Cigall, in your work, you have found that the remodeling of some of these complexes in greater than 20% of all human cancers appears to be fairly common. Can you outline what some of the key features of these structures are, and a little bit more about how understanding them more fully will lead to the ability to hit these as targets for cancer therapy?
Sure. So let's dig into these very complex, if you will, complexes a bit more deeply. So these are large molecular machines, actually 2 megadaltons in size. That's, if you take the DNA that's wrapped around each one of the core structures of chromatin called nucleosomes, that's actually 12 times the size of even that core structure unit of chromatin. So these are large molecular machines that are powered by ATP, a process called ATP hydrolysis, taking energy and moving these large structures up and down the structure of DNA to enable accessibility and to enable that timely and appropriate gene expression I highlighted in my introduction. These are multi-subunit protein complexes. There are multiple components that can come together. And to make these even more complex, they can come together in a combinatorial fashion. And each individual combinatorial assembly has a different effect on the genomic architecture within the nucleus of each cell. And so for years, these complexes have really been thought to just be to maintain our genomic integrity, to maintain the genome, to maintain DNA accessibility, and maintain proper gene expression. Never did we really imagine that these would come to the forefront of attention and be major therapeutic targets in cancer and other neurologic disease until sequencing efforts really told us so. So starting in about 2011, I was actually a graduate student at the time, and almost weekly in Nature Genetics and in Nature and every other journal came these studies that repeatedly implicated the genes that encode those various multiple protein subunits of these complexes in cancer. And this, for the first time, brought to our attention that perturbation to chromatin remodeling, this process called chromatin remodeling of these large molecular machines, was a major driver in cancer. And in some cases, the clear, irrefutably clear driver in very specific cancers, including pediatric cancers, which both our group as well as Bob at Epizyme have studied in great detail, as it opens a window to understanding some of the more common, more mutationally burdened cancers. And so our group has taken a combination of approaches, biochemical, structural, et cetera, toward really understanding what are the nodes we can target within these complexes. In some cancers, the subunit, or one component of these complexes, is completely lost. And so that begs the question, well, how do we approach a loss of function? How do we find unique synthetic lethalities that might represent viable therapeutic targets? And in other cases, there are gain of function mechanisms. This large molecular machine gains the ability to activate genes in an aberrant fashion, displacing repressive complexes that normally keep genes turned off. For example, those that are targeted by some of the drugs developed by both Constellation and Epizyme. And so it's this understanding of both gain and loss and even change of function that our group is eager to understand. And I think it's those nodes that provide the most viable strategies for therapeutics moving forward.
Thanks. Another area that actually has been alluded to in a couple of places is the idea that cancer is really a result of aberrant gene expression. And in many respects, some of the examples that people have already talked about demonstrates that. It's demonstrated in a lot of other products or a lot of other conversations and products that have actually reached the market. The thing that's a little bit more subtle is an area within it that's fairly unique to epigenetics and really involves the idea of how transcription occurs such that genes actually do express. And in the course of that, it leads directly to the topic that Jeannie has already brought up that I'd ask her to dig a little deeper into, and that is the area of long non-coding RNAs and how it leads or inappropriately leads to the expression of cancer. Jeannie?
Right, so long non-coding RNAs are defined arbitrarily as transcripts that are 200 nucleotides or greater, so they're distinct from the small RNAs, the pi RNAs, siRNAs, and microRNAs that you all know about. So, and these long RNAs exist in the nucleus to affect gene expression. And again, getting back to the point that I was making earlier, some 99% of the genome is non-coding, 90% of which is transcribed at some point in development. And to paraphrase what Nancy was saying earlier, it's this non-coding space that distinguishes us from each other and us from chimpanzees and healthy from abnormal cells or healthy from cancer cells. So what's going on in this non-coding space is extremely important. And I'll be honest that a lot of this transcription will be spurious, meaning non-functional, but we're seeing emerging examples of long transcripts that are clearly doing something to regulate gene expression. For example, there are transcripts now that are shown to be tethers for protein complexes or epigenetic complexes. There are non-coding RNAs that will scaffold together or bring together two different epigenetic complexes. And then there are long non-coding RNAs that will sop up, or they'll squelch away microRNAs from hitting their proper targets, or even squelch away proteins from hitting their proper targets. So long non-coding RNAs could be activators, or they could be transcriptional repressors. And actually, they do a lot of the same things that proteins can do. However, and this may be relating to why the question that you may be asking yourself, which is, why did they evolve if they can do all the same things? Why should we pay attention to them if they're all doing the same things? And I think it comes down to one property of long non-coding RNAs that, as far as I know, no protein coding gene actually will have. And that is that long non-coding RNAs can target protein complexes to a specific place in the genome, to one place in the genome. And to my knowledge, no single protein can do that. And the reason they're able to do this is because long transcripts are long. They have enormous sequence as well as structural complexity. And many of these long non-coding RNAs are actually only expressed from a single location in the genome. And so a lot of the transcripts that we study in X inactivation are those types of transcripts, exists, IX, and so on and so forth. And so we can think about long non-coding RNAs as barcodes, right, that will sort protein complexes to specific places in the genome. Okay, so I think there's going to be a great utility to looking at long non-coding RNAs for cancer, and we're already starting to see examples of how these transcripts can affect cancer development and cancer progression. And so I'll just rattle off a few examples. MALAT1, which is a highly abundant transcript that's been implicated in metastasis of lung cancers. Two beautiful papers recently showed that NORAD, this is non-coding RNA NORAD, and truncated forms of certain tRNAs will be upregulated during stress and will be important for suppressing genome instability. Okay, so we can think about them as sort of players, key players in tumor suppression. And then I'm sure you've all heard of these siRNA RNAs or ceRNAs that sop up microRNAs and thereby play very important roles in cancer progression. And then a transcript that we study in the lab called EXIST, which when we delete in the hematologic compartment of the mouse will result in a tri-lineage blood cancer with essentially 100% penetrance. And so female mice that lack EXIST non-coding RNA will develop things like myeloproliferative neoplasms, myelodysplastic syndrome, and chronic myelomonocytic leukemia, as well as a bunch of other things. So a tri-lineage tumor. Okay.
No, in fact, I think you're doing a great job of explaining why, in some respects, we haven't seen an emergence of epigenetic-related drugs, just because of some of the inherent complexities in the stages at which they're being investigated, and in which, in fact, their functions are being peeled back. But I'd like to turn to Johnathan for a question for you. It's almost a nature of cancer that cancer has a unique capability of co-opting normal processes. And some of the work you've done is that things like copy number variation, which is needed by cells to treat respond to hypoxia, nutritional deprivation, get co-opted by cancer. And that's an area that you've studied a great deal, particularly with respect to the emergence of resistant clones in cancer. Can you talk a little bit about how you see that playing out, both with respect to its inherent properties, but then with respect to an opportunity to treat cancer drug resistance?
Yeah, it's so we didn't set out to understand or identify how epigenetics affects copy number initially. I was in Yang Shi's lab as a fellow, and we discovered histone demethylases. We went on to try to understand their biology. My lab is really after trying to understand what the biomarker or the therapeutic index is for these and which type of patients. What we ended up finding is that instead of regulating traditional RNA biogenesis or transcription the way that we think of it, we found that they actually regulated how the DNA is copied and replicated. And we found this because the demethylase that, in particular, this enzyme that we were looking at is amplified in a lot of cancers. And what we found is that it actually associated with a region that is on the chromosome 1 locus. It's packed with pro-survival, drug-resistant oncogenes. And what's been known for about 10 years now is this region represents about 25% of cancer on 1q21. And what's very interesting about this is we consistently see it. It's also been seen in the case of other things like dihydrofolate reductase in the mid-60s when we realized that you consistently amplify it in pediatric leukemias, and this provides resistance. There's certain regions that the genome has a predilection to have happen. And so what we've been able to demonstrate is that there are factors, these epigenetic components, that are going in and using their associated interactors, their megacomplexes, their associated proteins, their friends, and they're able to generate these regions in a very strategic way. And what cancer has done is it's hijacking those processes. If I go back 5 years ago and I said to you that we could regulate copy number at a distinct locus and it wasn't just massive instability, you might have looked at me and told me I was nuts. The truth of the matter is what we're beginning to uncover is how epigenetics regulates this. Most recently, what Pat is alluding to is we demonstrated that hypoxia, 1%, which is physiological, important in fetus. It's important in the bone marrow. We know a lot of resistant modules are selected there. What happens is hypoxia triggers this process. It mirrors amplification of a gene, the demethylase. It stabilizes it. The natural biology, you stabilize it. It executes this program of drug-resistant or survival genes. What has cancer done? What cancer's done is just hijack the process. It hasn't come up with its own novel system. It's not brighter than the average cell. It just knows the proteins or the pathways related to epigenetics or the construction of your genome so that it can do this. There's another key feature. These copy control events in the genome are in fact transient. They're not stably integrated. We can recapitulate this and look in tumor databases where we found it. We can look in any cell. The nice part about this is the phenomena that we're discovering about epigenetics and copies or the resistant selection does not bias to one cancer. It works across the board. Lung, ovarian, breast, pediatric, adult, heme, solid. And what this means is it begins to understand that the genome, pieces of the DNA are being selected, kind of like an extra suitcase. Imagine if you go to an airline and they tell you, hey, you can take this extra baggage on. You get very excited because that means you can bring presents back, or you can have souvenirs, or you can have extra clothes or whatever you need. That's exactly what your genome is doing. It has the ability to pick the regions it needs, and inside that are the genes that are innately providing a benefit. And there's a package of them at distinct regions. And so what epigenetics, or what we've begun to uncover and realize, is there's a whole connection of these factors that control that DNA environment that are controlling these events. And these are the drivers of the heterogeneity, and these are affiliated with those resistant selection. So imagine if you inhibit it. So what we were able to demonstrate through metabolic change, if you change the metabolic state, or more importantly, use small molecules, tool compounds at nanomolar doses, you can shut this down. So drug-resistant oncogenes important for myeloma, important for ovarian or breast, they disappear. You can reduce them. So it's a DNA dosage change, which in turn allows selection of those resistant modules. So there's a lot of plasticity. And that's the whole basis of epigenetics, is plasticity, which is the benefit of all of the therapeutic opportunities we have, because you can rheostat the genome. You can rheostat the genes. You can rheostat the process.
In fact, I think that's an excellent segue into the next class of discussion we want to have. I think it's very clear from these descriptions that epigenetics represents a richness of targets that have not yet been touched, or at least have only begun to be touched with respect to treatments of cancer. And so we want to turn now to the process by which drugs are being developed with respect to these kinds of targets and how these targets are going to be exploited. Nancy, you have done a lot of work, and Syros has done a lot of work around the concept of switches that control gene expression and a particular class of switches called super enhancers. Can you describe that a little bit and talk further about how that represents a putative target that would be useful in addressing certain kinds of cancer?
Sure. So, really amazing discoveries have been made over the last several years. A lot of these were really through developmental biology research. And there was a fundamental question about, why with the same genome do you get cells with completely different cell states and programs? And an area that's very important for that study are the enhancers. So think about the enhancers as the switches for genes. So each gene has its own switch. And by analyzing the non-coding region of the genome and looking sort of where the transcriptional machinery is binding, you can measure enhancers. So any given cell, you have 22,000 genes. About half of them are expressed. So you have about 10 to 12,000 enhancers, which you can measure through a variety of novel genomic technologies. But it actually turns out that there's only a small number of enhancers that are critical for the gene control program, for the cell state of a cell. And you can distinguish those because they're massive in size. They're like tenfold the amount of transcriptional machinery and expands over the DNA. And there, the term is called a super enhancer. It's just a large enhancer. So there's about a couple hundred of these super enhancers in each cell, and they control the master transcription factors and the professional products that make a cell what it is. And so in cancer co-opts that to make the expression of genes that are important in the hallmarks of cancers. So if you look in a cancer cell, you'll see super enhancers controlling proliferation genes, anti-apoptotic genes, immune avoidance genes, transcription factors like MYC that are oncogenic. So just by that, by looking at where those super enhancers are, you can identify the core set of important genes that are functional in a given disease. And the approach that we're taking at Syros is to use that to identify novel points of intervention in disease. So one of the focuses that we have is to actually go after targeting components of the super enhancer itself. What is the super enhancer comprised of? It's comprised of transcription factors that bind to the DNA, and then other regulatory proteins that bind to those transcription factors. And we and others have shown that by targeting components of the super enhancer, you can then downregulate a whole program that's important in disease. So rather than going after a signaling mutation or pathway, you can turn down a whole set of really important genes, such as in cancer. The other approach that we're using with this platform is to say, well, wherever a super enhancer is, it's controlling a gene that's going to be very important in that disease, in that cancer. So we can use that to point to protein coding genes that we want to actually target that could be very important. And actually, through that work today at the company, we now are taking into the clinic 2 novel drug programs. One is to a transcriptional kinase that's part of the super enhancer complex that's important in turning down a whole set of transcription factors that are oncogenic. And as you heard before, targeting directly transcription factors has been very difficult because they tend to not have binding pockets. But by lowering the level of expression of the transcription factors, we can actually have a dramatic effect in certain tumor cells. The other approach that we're taking is using a super enhancer to identify a novel disease dependency. And through our platform and working directly in primary human disease tissue, we've identified subsets of cancer patients that have novel dependencies. And we actually identified one to the retinoic acid receptor alpha, where there was already an existing drug that we were able to bring in that was never developed in this subset of patients that we now have brought in, and we'll be going right into a phase 2 clinical trial in a novel genomically defined subset of patients based on this work. So I think you can understand that by studying and analyzing these really important critical regions for controlling the gene program, there's multiple novel approaches and targets that one can take for helping patients with a variety of cancers.
Continuing on in that same point, Keith, one of the things that when you think about the kinds of targets that you're facing in epigenetic and epigenetic regulators that you want to target from the point of view of cancer therapeutics, is it practical to think about following the normal drug development of high throughput screening, medicinal chemistry, lead development, early clinical candidate development, or do you need a different paradigm for drug development in order to exploit some of these targets?
So Pat, that's a great question. It's where the rubber hits the road in terms of any new targets. Let me step back to see if there are two key things hopefully that people catch from this panel. One is what something Nancy just said, which is what we're trying to do with epigenetics is to turn on and off programs of genes. It's not a single gene that tells the cell to multiply and ignore all the stop signals. It's a program of genes that tells the cell what to do. And the second part of it is this druggability question, Pat, that you're asking, which is can we drug these targets? And I think up front, there's some of the classic issues that are there. There's the question of, can you get bioavailability? Can you get cell permeability? Can you get nuclear permeability? Can you get potency and selectivity across these different areas? And I think the answer we're finding out is yes, you can, but it's different for different classes of targets. And we faced this question early on with Constellation when we decided to go after readers, writers, and erases, 3 different classes of molecules. And we've learned since then that it's a different answer for each class. If I look, for example, at bromodomains, which are acetyl readers, what we found is a lot of the domain structures are very similar. So structure biology helps us dramatically in terms of developing molecules. And we can also target hop from one target to another. And so we've developed libraries that are potent against one molecule that give us good initial hits against other targets, and we can go through that. Now, what we've learned with that is we have to focus on selectivity, because once you're target hopping, you've got to then select out the various different targets that you don't want to have with any particular drug. If you look at another area, for example, methyltransferases, EZH2 being a classic example, I think Bob and I could both say they're hard. You don't get target hopping, you have exquisite selectivity, but it's very hard to find molecules that are actually potent, bioavailable. In fact, EZH2, a target that the industry has been going after for a long time, there are only 2 bioavailable molecules that are in the clinic, and they were sitting right next to each other in terms of the 2 companies. But it's a hard process with very intense medicinal chemistry to be able to zero in on those targets. And structural biology historically hasn't been important. It's just starting right now to see the first structural support coming down in that direction. Demethylases, I'd say somewhere in between. The molecules that take away methyl marks, for example. We see a certain amount of target hopping, but we also see selectivity. We see a classic enzyme type approach, if you will, on some of these. But we use both of those tools there in terms of more of the traditional medicinal chemistry, screening, as well as target hopping to get there. And I think the key thing we're finding is that these novel biological targets that are being represented across the table here are in fact druggable. It takes work, takes different techniques, specialized techniques at times, but we do have potency, we do have selectivity, we have bioavailability, and we're now seeing in the clinic the early signs of efficacy for them, so it's possible.
Bob, you're the perfect person to continue with that. Now, you've been doing enzyme inhibitors on several companies for a large number of years. And in fact, I think Epizyme had published a list of about 90 targets, which are HMTs by and large. But how has that developed? How has that been progressed? In a similar kind of question regarding druggability, are these classes of targets really accessible with respect to the ability to develop drugs?
They absolutely are, Pat. Thanks for the question. I think that Keith touched on some of the key points already, but I'll elaborate a little bit. First of all, given my choice, I would always choose to go after an enzyme target because the biochemical activity of enzymes and their structures just lend them well to modulation by the physical chemical properties of small molecule drugs. And so at Epizyme, we focused right from the beginning on a specific class of enzymes involved in chromatin remodeling, the histomethyltransferases, and we were very narrow in our focus for what I think are very good reasons. Almost 8 years ago, we understood that some of these enzymes played a pathological role in human disease, that they had active site structures that looked like they were going to be amenable to small molecules, and they looked like they created opportunities to have a diversity of chemical structures and binding modalities with which to inhibit them. And that's all worked out quite well for us. Our first-in-class EZH2 inhibitor, for example, tazemetostat, has now been in the clinic for quite a while. It's in multiple phase 2 trials. We've had our DOT1L inhibitor, pinometostat, in phase 1, and with our partners GSK, we hope to be bringing a third compound into the clinic shortly. The success of that work, I think, has really been driven by pretty traditional drug discovery, a strong emphasis on understanding the biochemistry and the physiological context in which these enzymes work, very strong medicinal chemistry. I'll differ with Keith just slightly and say that for Epizyme, structural biology has been incredibly important, and we've solved close to 800 crystal structures now and have used those to drive compound optimization. And the final thing I would say is that not all enzymes are created equal in terms of being good drug targets. What we look for at Epizyme are situations in human cancer where a genetic lesion, either at the target itself or in a related pathway, leads to an addiction of that cancer on the enzymatic activity of that target. That provides us a patient stratification mechanism. It also provides us a mechanism for a large therapeutic index. And again, those genetic lesions may be directly at the target, but in the case of EZH2, one of our phase 2 trials is in INI1 negative and SMARCA4 negative solid tumors. Those are both subunits of the complex that Cigall has championed, the SWI/SNF complex. So there are situations where loss of function mutations at one protein create an addiction of the cancer on a very different protein, and that's what we're exploiting at Epizyme.
I think one of the, and we're getting down near the end of the session, but one of the observations as I listened to the presentation this morning and the movement from basic biology into drug development and even into the clinic is it strikes me that while historically this hasn't moved, we're only now beginning to see drugs in the clinic. It may be epigenetics is in a position in which basic biology can be translated to intervention more quickly than we will see other classes of targets or other kinds of compounds that we're used to seeing a drug development cycle that's of a certain periodicity. But is it likely that, and I'll throw this to anyone, that what we're going to see over the next 10 years is an acceleration of the movement from basic biology into drugs, into the clinic, or is that just a hope?
So I'll just take that from the demethylase point of view. 2004 in December is when lysine demethylase, or LSD1, was discovered in Yang's lab. At that time, from that time under 10 years, we went from protein discovery of a new enzyme, new role, to the first phase 1's happening in Europe. It's under 10 years from gene, novel function, all the way to bringing it there. The lysine demethylase is this class, these KDMs, these Jumanji proteins, discovered in 2006. Now, we're on the forefront of these about to hit the clinic space with various groups. So we're talking under 10 years we've been able to take it that far. So I would answer yes, I think we can do things very quickly.
With that, I want to thank the audience for your attention, but most importantly, thank a very educated and very articulate panel describing a very complex subject, and I appreciate very much the ability to tie these together as you have. Thank you.